go. Welcome to another bi-weekly gathering. I'm going to hand it back over to McFinn. My name is James, and I, along with Grace, who you've seen here in the last 30 minutes, we're going to be moderating some of your questions from our presenter today. But I'm going to give it right back now to McFinn.
Oh, there you got it, folks. It takes 2 weeks for me to get to be with you guys. Sometimes that's a long time. It's like, Terry, when you smile at me, it doesn't go away. I remember those smiles. And each of us has something that we all bring, even if we're silent. Do you know that your presence means a lot and it says a lot? So thank you to each of you for showing up, because man, I ran up the hall. I was like, I'm going to be late. But you know what? None of us are ever late. Don't ever think you're late. As long as you're on the way, we'll get there. Okay? Together. With that, I'll turn it back over to you, James.
Thank you, McFinn. Tonight, we've got a very exciting presentation for Dr. McGrath. I'm going to read a little bit of an intro before I turn it over to him, since he is the expert. Dr. Michael McGrath is a professor, departments of laboratory medicine, pathology, and medicine at the University of California, San Francisco. He has authored over 200 scientific articles in the fields of AIDS, cancer, and neurological disease since joining UCSF in 1985, and has reoccurring NIH funding to support his research programs in excess of $1.5 million per year. Dr. McGrath has been a pioneer in the recognition of the role that inflammation, and especially macrophages, play a role in multiple diseases. His recent focus has been on the study of macrophage activation in the pathogenesis of ALS and other chronic diseases. Dr. McGrath maintains an extensive resume in HIV and oncology as well. His career in HIV began early in the HIV epidemic as assistant professor of medicine, AIDS Oncology Division, University of California, and San Francisco General Hospital. He also began serving as an ad hoc grant reviewer for the NIMH, NCI, NIAID, and the VA, and joined the Scientific Advisory Board for the American Foundation for AIDS Research, where he continues to serve. We are very excited to have Dr. McGrath with us today, and with that, I'm going to turn it over to you, Dr. McGrath.
So McFinn, I love the name. And I like the beard. You know, this is my COVID preventer. You know, I've got my mask on permanently. So, well, it's a real pleasure to be invited to give a speech or, you know, to talk with you all. I've tried to gear this presentation towards a way of encouraging discussion as we go along the way. And I think I'll start off by showing this very weird picture of me. Where is it? There we are. I think it was photoshopped. I can't really tell for sure. Yeah, let me fix one thing. Just a minute. I have to fix this. How can I not fix this?
If you can just hit on that bottom right of your screen, that little presentation button, it'll make it so much bigger for all of us.
I know I did, but you know, I have two computers going here. No. Oh. You know, I don't. So I have a separate computer, and it won't let me put it on presentation mode for some reason. I just turn everything on. This is terrible. I need my own AV person. So
if you go to the page before, I think I can help you navigate.
Okay, so so what's happening is I've got two computers. One of them is a monitor that I'm looking at. The other one won't let me change it to a, normally it lets me do this.
I guess
The
this should be
presentation
fine.
mode.
We do see enough of your slide.
Yeah.
No, it's
Yeah,
terrible.
this is
though.
fine.
Oh my goodness, it's not fine though. I just think so bummed by this.
Oh, that's a great
Um,
solution. SUK. If you go to the display
Can you s-
setting,
You're on top.
yeah, I think
display.
if you ended the screen, if you get on the top, you see end slideshow, perfect.
Okay,
And then will you share your screen again so I can see that?
so there's screen 1, screen 2. So the problem is I go to this and it puts it in a presentation mode. My
Yeah, there you go.
monitor is got it
Perfect.
It's
full.
okay. It's good.
See, but you can't see my pointer? No,
We can see your screen.
the screen, but no pointer, right?
No, there's no pointer.
Jeez. Oh my God. You know what? I have way too many degrees and not enough practical knowledge. This is really something. This is really something. Let me try one more thing. No, that didn't help.
If you Nitesh is giving us suggestions. Thank you so much, Nitesh. If you right click, you can get your pointer. So end the slideshow and then, oh, that worked. And then
There it
right
is.
click and you can get your pointer. Oh,
Right click and I can get.
oh, there you go. You have
Hi,
your
presenter
pointer
view.
now.
Oh, there we go. Does that work?
Yeah.
Oh my goodness. Who's the
Natasha's a
genius that did
genius.
that? All right.
Thank you.
Okay. Gotta go to the school with you. This is awesome.
He's an ALS researcher, so thank God for that.
Yeah,
So
thank you so much.
this is awesome. So I just want to give you just a teeny bit of background. So when I was being introduced, you know that I have a lot of experience working with AIDS. And back in the day, there was something called AIDS dementia that was so profound that there were hospices that were filled with these fully demented people. And it was 100% disease of macrophages that tracked into the brain, that went from the blood into the brain. And the severity of the dementia was related to how many inflammatory macrophages were in the brain. And so when antiretroviral therapy came along and the virus was no longer making all these weird side byproducts and the macrophages no longer went to the brain, people woke up. There were people in the hospice that woke up and went back to work. And I said, wait a second. Maybe neurologic disease is more complex and more interesting than we used to think. And if you look at neurologic disease, and if you just think of Alzheimer's, very long and slowly progressive disease. And the notion is that you can't really reverse neurologic damage once neurons are dead. But the question is, are neurons really dead or are they just dysfunctional? And so that's how I came to start working on ALS in 2001 with Bob Miller at CPMC in San Francisco. So let me just, there's this weird little thing up in the front. And, all right, so as much about ALS as I do, really. You know, it's broken out into sporadic and familial. There's been a lot of work on genetic modifications that one can find in a minority of patients. You can see that there's a lot of work that's been going on in TDP43, trying to figure out how it actually causes damage or dysfunction to the brain cells. And then there's inflammation, and we're going to talk about that today. And the question is, what type of inflammation are we looking at, and how can we identify it, and then how can we reverse it? And of course, there's a lot of therapeutic approaches that people have taken for the last 20, 30 years, mostly, unfortunately, failures. And we're going to break that down a little bit too, and try to figure out maybe the best ways of trying to figure out how drugs may be beneficial. Now, this is a neuron. Not really, it's a picture of a neuron. But so if you look at the neuronal body, you can see where almost all the research and the abnormalities are focused on. So you see TDP43 aggregation, you see mutations in C9orf72, SOD1 aggregates. So you've got issues with protein aggregation, glutamate excitotoxicity. This is a receptor system in a process that really is all it's thought to interact with. You've got factors that are impairing the glutamate uptake. You've got mitochondrial dysfunction. This happens to be one of the targets that Amylyx drug is supposedly targeting, be able to supply. This is the energy generator within a cell, the mitochondria. And then you've got, as cells die or become dysfunctional, they elaborate all these neurofilaments from the axons. And so you know that people are starting to look at neurofilament as it falls off because the axons are dying. And of course, then they've got microglia in the cells, which are the brain macrophages. And in certain circumstances, they are elaborating things like a reactive oxygen species. Of course, the darabone is directed against those sort of things. And to date, really targeting only the CNS, it's failed really to halt disease. I think, you know, we've got a little pieces of activity and hints of ways to be driving the bus going forward. But so far, this is a snapshot of where most of the efforts of research and therapeutic development have been focused on. And so this is the title. I don't know why I came up with this title. This is a systemic immunity. So I just got my flu shot. And of course, it's supposed to be inducing systemic immunity. Well, immunity means different things to different people, to different parts of the immune system. So the question is, ALS an inflammatory disease? And if so, we need to really describe it. And so I put everything down on a whiteboard and took a picture of it, and I figured it out. And all you have to do is, well, wait a minute, maybe I didn't really figure it out. But the point here is that if you look at immunologic reactivity and inflammation, it's very complicated. And so the challenge that we have really is to simplify approaches so that we can actually achieve something. And so let's understand inflammation at a real practical level. So when you get a wound, you get neutrophils that are the really short-lived white blood cells that make up most of the white cells in your blood that attack the bacteria. They can form abscesses. The next type of inflammation involve lymphocytes, and these are the things like COVID, for example. So viral infections, intermittent inflammatory states, multiple sclerosis, inflammatory bowel, rheumatoid arthritis. That's this type of inflammation. The type of inflammation that is involved in chronic diseases that last a long time, and that really involve the central nervous system, AIDS, dementia, Alzheimer's, ALS, Parkinson's, FTD, and then genetic diseases where central nervous system symptoms. And so you've got a mixture of different types of inflammation. And it's really important to understand what type of inflammation is involved if you want to try to address it therapeutically. So this is obviously it's a very simplistic view, but just imagine the timing on this. You get a stab in the hand. You can see here we go. There's a pin, and it causes a lesion. And then your immune system, and this is now the innate immune system. This is something that all animals, all the way back to multi-cell organisms, will respond to a wound this way. And so there are tissue macrophages. They're the first things on the scene. They recognize all this damage and they make factors that call cells from the blood. And then these blood cells go and react against the wound, and you develop a clot, and then this heals. This takes a week, 5 days to a week. When I got a vaccine, or you get your COVID vaccine, how long does it take for that vaccine to kick in? It takes a few weeks. So the point here is we're looking at two different types of immunity, one that really works pretty acutely. And then the other, which takes a long time. And it turns out that the macrophages in the body are involved in this acute reaction, and they're really the fundamental defense system of the body. And so when we think about, I wonder if I can, oh, I can move this thing. All right, hang on. Can I move it out to the side? Oh. All right, so we're talking about the innate immune system. So when we talk about macrophage-associated diseases, we're talking about an innate immune system. The innate immune system is very old. It's primordial immune system where monocytes in the blood, not granulocytes, these are the short-lived phagocytic cells, but the monocytes turn into macrophages. They can also turn into dendritic cells, and these are the ones that tell the adaptive immune system what to do. These are the ones that tell the T cells and the B cells, to make immune responses against the COVID vaccine, for example, or viral elements. And so this is the component of the immune system we're going to be talking about, which involves macrophages, and it's a rapid response, a part of the immune system. Somebody needs to be admitted. It stopped functioning. Oh, there it goes. All right. So I don't know why I just stopped. This is where all the macrophages live in your body. Shocking. 15% of almost all your organs are macrophage. Some of these are quite long-lived. The macrophages in the brain are called microglia, and they turn over maybe 1% per year. The macrophages in the spleen maybe turn over every few months. Macrophages in the liver turn over maybe every 6 weeks or something. But the point is that these are very long-lived cells, and the tissue macrophages live a long time, but the bone marrow-derived macrophages are like the cavalry, in that they come out of the blood as monocytes, and they can traffic everywhere in the body. And so these are the bone marrow-derived macrophages are the ones that run around the body in the blood looking for things to do. And so if you look at how, in a very simplistic way, how I believe and disease works is that normally you've got macrophage activation that's balanced. So remember that 5 days of a wound, you have inflammation, and as the inflammation starts, think about the wound being red initially, and then starts clearing up. It's because of this feedback loop that you've got a wound healing macrophage that makes factors that then balance this out, so that you've got this self-regulatory loop going on in all of us every single day, every microsecond. Chronic disease, though, you now have an imbalance. And so now you've got too many of these inflammatory macrophages and not enough of these wound healing ones. And so this is the challenge. The challenge is to figure out how to identify these cells and to fix them. There it goes. Right, so I'm just going to tell you that I just didn't invent this. This is the G990 3A mouse, which is the SOD1 mutant. And you can see as they become symptomatic, these blood macrophages come from the blood into the spinal cord where there's disease. They don't go everywhere. Here's the brain from the same animals, and they aren't really going there. So the point is that you're having blood-derived macrophages going into the spinal cord, and they're the ones driving disease. Stanford showed the same thing in humans. It's a critical experiment that was done quite a while ago. I can't move this thing either. Where what he did is he took ALS patients and asked the question, are the macrophages driving disease? Are they bad inherently? And if I can replace them with good ones, will the disease stop? And so he got allogeneic bone marrow that was from normal people, and he transplanted them into ALS patients And unfortunately, that didn't do anything clinically. But what he did find out is that when the patients died, the average about 6 months after the bone marrow donation, more than a third of all the cells were from allogeneic bone marrow donor. So think about that for a second. All these brown things are macrophages. They shouldn't be there. This is the spinal cord motor neuron loss. This is the ventral horn of the spinal cord. And all these brown things are these inflammatory macrophages. And many of them are from the bone marrow donor. So the point here is that you've got an aggressive migration of blood monocytes into the tissues that are continuously driving the inflammation. Now, I'll tell you that this is a subset of patients. And as you saw from the very first slide that I may or may not have shown, that ALS is a heterogeneous disease. And I think it's important for us to be able to establish what the active part of the disease activity is that we're going to go after with any particular drug. I'm a cancer doctor as well. And if you were to walk in the door and say, I'm going to cure cancer, they'll say, what kind of cancer are you going to cure? Breast cancer, lung cancer, bone cancer? All the cancers are different. And as you know, the cancer therapy development has been focusing on different pathways for different subsets of patients. And I believe that ALS is moving in the same direction because it's now recognized the heterogeneous nature of the disease. And the FDA is even in their guidance document from 2019 recognized it as being heterogeneous and that drugs should be developed targeting specific subsets. Now, just because you've got macrophages in a tissue doesn't make that tissue necessarily good. This is a big VA study, and you can see the bone marrow, the spinal cord, somebody who had a 10-year course of disease here on the right-hand side. On the left-hand side, this is a patient who had about a 2-year course of disease. You see all these brown cells are macrophages. And so high density macrophage, even though they're quote unquote wound healing cells, is a bad thing if they're making factors that cause your central nervous system dysfunction. Oh, I see what I was doing wrong. So what are we going to do about this? What I'm going to talk about today really is what we've been doing. When I say we, it's Neuvivo. It's a company that took over the development of MP001. You may have heard about it. A company called Neuraltis was developing this for about 10 years. Neuvivo, and I'll introduce the people in Neuvivo at the end, took over all of the databases and tried to figure out what happened in the development of MP001 to sort of, I guess, interfere with its development. And what I wanted to present today is some of the things that we've discovered by looking at data, both from the 2 phase 2 clinical trials, as well as performing some new biomarker analysis that actually really illustrate how the drug works and actually how a subset of ALS works too. So very simplistically, I was showing you pictures that monocytes in the blood can turn into inflammatory macrophages and they traffic into the central nervous system. And that this process is pro-inflammatory. The nuclear factor kappa B is involved in a subset of these things. And once you've got cells in the central nervous system that are making factors, they suck more cells from the environment back into the tissue. And it ends up being one of these self-fulfilling ongoing processes which you really can't interfere with to date. So what are we going to do about it? All right, so this is, oh gosh, all right, put it over here. Okay, so this is, I'm going to give you a cartoon of what happens in inflammation. What happens at beginning, let's say if you've got a macrophage, it comes in contact with bacteria. This bacteria is killed through a process called an oxidative burst. This produces hypochlorous acid as a byproduct. Of course, this is very toxic. It kills the bacteria. Within the phagocytic cell, it's got an amino acid called taurine. And now you make taurine chloramine, which neutralizes this acid. But more importantly, it makes a factor, taurine chloramine, that now converts these inflammatory macrophages to a wound healing form. And this taurine chloramine regulates NF-kappa B, among other things. So it turns off inflammation. And this cell then makes factors that then cause resolution of this process. So this is this self-regulating cycle that is the central part of the innate immune system. These are very long-lived, can be very long-lived cells, and they are self-regulating, except in the context of disease, where you've got many of these. So what are we talking about? Turns out NPO1 is sodium chlorite. So it's relatively inert unless it comes in contact with a heme-associated iron molecule, and then it's converted to taurine chloramine. Whoops. So sodium chlorite converted to taurine chloramine, and taurine chloramine is a very long-lived molecule. So as you may or may not remember, the drug is given, you probably don't remember. I mean, the very first phase 2 clinical trial was in 2011. Second phase 2 trial was in 2017. We have all the data and all the databases from those 2 trials. But basically, this drug is given in 3 days, 1 hour, half-hour infusions, and then you stop. It's because this molecule regulates so in a strong way. And so that's the basis for the intermediate treatment program. Now, this is the paper that was published earlier this year. And what we did is the phase 2B, which is second phase 2 trial, had never been published by Neurologist. And so we looked at the data, or Neuvivo looked at the data, and reported the results of the phase 2B clinical trial. And none of the primary endpoints were achieved. And so we started looking more and more and more at post hoc, of course, because we didn't run the trial. So looking at all the data and with one premise in mind, and that is in the first phase 2 trial, there were a subset of people, about 25% of people, who did not progress at all over the period of the 6 months. Now, it's clear that ALS patients can have windows of time where they don't progress either. But in the case of the first phase 2 trial, it looked as if there was a dose response in this particular halting of progression. So we just asked in this study, this phase 2 study, if we just looked at the people who stopped progressing, who were they? Well, it turns out they had this inflammatory molecule, C-reactive protein, was greater than 1.13 milligrams per liter. And this represented about 76% of the patients in the study. So it's not a small proportion. CRP, I'm going to talk about CRP in a minute because I think you'll find it fascinating. The other thing is that it turns out that you can probably just have this younger than 65 or 65 and younger, because people who are older than 65 have got a lot of other things going on, heart disease, cancers, things like that, or arthritis, stuff that gives you high levels of CRP that maybe has nothing to do with ALS. And so that ends up above 65, the outcomes are noisy. It's not that, that MP001 can't have an effect against people that are older than this. It's just that for the purposes of the clinical trial, it gives you a noisy result. And so let me talk about CRP for a minute. So C-reactive protein it used to be thought of, well, in fact, people still think of it as a non-specific molecule that's turned on in the face of inflammation. So let's look at really high levels of CRP. Bacterial infections, awesome inflammation, rheumatoid arthritis, very inflammatory. And this is the CRP levels along this particular axis. Cancer has got inflammatory components to it, viral infections, And then we now start talking about the usual things, atherosclerosis, obesity, etc. And it's mostly been used to follow people with atherosclerosis. Levels of above 3 are associated with higher rates of atherosclerosis. And we're looking at a cutoff point here of CRP with 1.13 milligrams per ml. And I want you to have no opinions about CRP at this low level until I just show you one thing. CRP is real. It's a real primordial immune system molecule. It's actually really amazing. This whole notion that is just turned on in the face of inflammation or infections is not true. It's actually an effector molecule. So this is a pentamer that binds to an infected cell or a dying cell and allows then the macrophages to more efficiently either kill an infected cell or to engulf it and recycle it. So this is like a primordial antibody almost, and that it helps the immune system, the innate immune system, function. So our thought about what CRP is doing in ALS is that there's a subset of people who are trying to get control of their ALS, and they're making this CRP to help clear damaged cells. So that's one way of thinking about it. If you look at in our phase 2, the 2 different phase 2 trials, and this is now what I call stabilization rate. These are people who have been on the drug for 6 months and have had no loss of ALS function rating scale at all. And this is in the entire 2 phase 2 trials, and people who started at starting point, of course, and finished after 6 months. And you see that people who had greater than 1.13 CRP, who got the drug as compared to placebo, highly different clinical outcome. These people have about 35% non-progression. These people are about 11%. If you have a higher CRP, these are people who start off with 3 milligrams, there's a 10-to-1 difference between drug treated and control. So this should tell you if you're a hunter of a mechanism, like how can this happen? This is, somebody once said to me, well, if you have a bag full of half black marbles and half of white marbles, you can sometimes get about 5 or 6 in a row, or maybe 10 in a row. They're all black or all white, and it's random. But so how are you going to know whether this is random, this difference, or not? Look at the statistics, tell you it's not random, but we have to take it to the next step and say, what's a potential mechanism? How can this be? So one of the other effects that's actually quite profound. First of all, the ALS functional rating scale was halted in that subset of people. And this is actually really amazing finding. And this is the vital capacity change from baseline. And of course, this is slow vital capacity, or it's from the phase 2A, it's converted forced vital capacity. But you see in the drug-treated arm, after 1 to 2 months, now we see that there is a change in the slope of loss of vital capacity function, whereas the placebos are progressing at the same rate, which is losing about 2.5 units of vital capacity function, units of percentage per month. We see that the vital capacity is slowed, loss of vital capacity is slowed by 50%. And there are no drugs that are out there right now. Of course, BiPAP and ventilatory function can give you something like this, but absent ventilatory support, there's nothing that has been reported in the literature in any clinical trial that's had this sort of a magnitude of an effect on vital capacity. So now we've got this subset of people who stop progressing. We've got people whose vital capacity has improved. How can that be? So I'm going to give you a theoretical basis of how this can work. There's something called microbial translocation. It was first defined in AIDS back in 2005. In the face of chronic inflammation, let's put this in the context of ALS. In that we all know that there's something happening in neurons with ALS, whether it be misfolded proteins, or you can pick your favorite abnormality, which is annoying to the brain macrophages. These make factors that then can cause systemic inflammation. This inflammatory cell, remember, inflammatory cells can't eat. An inflammatory cell makes factors that cause dysfunction, but they don't eat. So what's the problem with that? Well, it turns out that you see in the colon, quite a large organ in all of us, that are filled with what? Bacteria. The colon cells that make up the wall of the colon turn over every 5 days. Billions of cells turn over every 5 days. And if your macrophages are not eating to protect you from these bacteria, what you have now is a leak of bacteria into the colon, and now you've got these bacterial products. Lipopolysaccharide is a major bacterial byproduct, and what it does is causes more macrophage activation. This macrophage now is called by the brain or the spinal cord saying, oh my God, we're damaged, we need help. And now this cell comes from the blood into the tissue. It's already activated. It has no way of helping. And so now just imagine this loop going on and on and on. This is what's called microbial translocation. And so far, well, I don't know that there's a way of really stopping this other than there are ways of healing the colon. But the point is that this is a shared feature in ALS, Alzheimer's, Parkinson's, other central nervous system diseases where there is this quantifiable lipopolysaccharide in the blood. If you can measure this in the blood, it means that there is this process that's ongoing. The good news is you can now measure this and try to do therapeutic interventions to say, am I affecting this molecule or not? If so, do I affect this process of a microbial translocation? Sometimes it just doesn't want to work. Okay. It's trying to go. Okay, so we did a biomarker study, and I'm going to tell you about this. The biomarker study that we did, the rationale for it was to look at specimens from patients who had a positive clinical outcome. In other words, we're looking back at the phase 2 studies that were done by Neuraltis, and we wanted to look at people who did better and say, what are the characteristics of people who do better as compared to controls? And is there, if they do better, is it because microbial translocation was affected? It could be because of something else. But we designed the study to be able to test whether microbial translocation was involved. So we looked in vivo. I mean, these are real patients. These are baseline blood values compared to blood values at the end. We've done a lot of studies in test tubes and in animals. And of course, they're good for test tube and animal reports. What I'm going to show you is data from humans, ALS patients who had received the drug. And in this case, with NPL1, proposed MOA, as you know, is as a macrophage regulator. And so in theory, if it reestablishes the innate immune system balance, it ought to be able to fix this inappropriate activation and make those cells then phagocytic and reestablish balance. There we go. Okay, so as I said before, it's shared between all these different neurologic diseases. So the patient population we looked at were the phase 2A NPL1 patients who had Biomarker, had plasma specimens stored frozen at minus 80 degrees. And we looked at baseline values and compared them to 6 month specimens. But we knew the clinical outcome of these patients on a month-by-month basis because they were involved in the clinical trial. So this is the ALS FRS change from baseline. And this is the number of months. And so these patients had a positive response in their ALS functional rating scale. They also had a positive response in their vital capacity. So this patient population is the one we studied, and we looked at baseline and then follow-up values for a variety of different biomarkers. So just an example of a couple of the biomarkers, interleukin-10 is a very potent suppressor of inflammation. So it's one of these, remember I told you the cyclical process that goes on where you've got inflammation and then anti-inflammation. Well, IL-10 is one of these anti-inflammation molecules. And so it's turned on when you're regulating the immune system away from inflammation. So you see that at baseline, as compared to 6 months, the IL-10 level went up, and in the placebos, it went down. In this case, LBP is something called, LPS binding protein. And this is a surrogate marker for LPS. This is the difference. If we look at plasma level decreasing, these are factors that went down. So all of these factors are promoting abnormal function. They all went down, highly significant. These are factors that went up that promote wound healing, like epidermal growth factor, neopterin, IL-10. These are all factors that promote wound healing. There are factors that didn't change at all. IL-8, TNF-alpha, interferon, and IL-6. They were normal at start in this patient population and did not change. And so it ends up now looking like this. This is the take-home message of how we think the drug works. And then it starts off, this is the microbial translocation. With NPL1, we turn on wound healing factors. We turn off pro-inflammatory mediators. Remember how fast the innate immune system works? This takes a week. So this process takes a relatively short period of time, and you can measure it very simply with looking at factors like this. Now, there happens to be, you heard from another, one of our colleagues, Mike Snyder, who's done some elegant work. And in the last 2 years, it's been very clear now that ALS starts in the periphery and actually interfaces with the peripheral immune system at the synaptic junction. So from our perspective, this is something that the blood macrophages interact with on a daily basis. And you know that as you cause demyelination, you get distal axon degeneration, and you get production of neurofilament, light chain, heavy chain, things like that. MPL1 regulates taurine through taurine chloramine. These factors regulate a process that's systemic. We don't know, although we're in the process of testing, whether there's a change in brain-related material, as has been reported recently from Biogen. But this is sort of a summary of where we think our drug works. Can't do it right. So this is the picture. This is the neuronal body. A whole bunch of different abnormalities have been mapped here. The drug's ineffective. We have to keep trying no matter what. Some of these factors have already been approved for drugs. Others, for example, like Amylyx drug that's being looked at right now, maybe targets the mitochondria. We're taking a completely different view. We're looking at interfacing at the periphery, the blood macrophage mediated interface. We've got blood markers that track with clinical outcome, and we've got a way of determining a drug activity. And so let me just finish up by saying this is a small molecule regulator, first in class drug. It's a disease modifying anti-inflammatory. We know the mechanism of action. I've been able to show it to you today. We have statistically significant improvement in 3 different areas. the ALS functional rating scale, the vital capacity, and a subset of people with stabilized disease. This first paper was published in Muscle and Nerve earlier this year, and we're in discussions with the FDA right now. My partners here is Year. It's one of my co-founders and the CEO. Vern Norville is our other co-founder. He's involved with all the intellectual property, probably the most amazing intellectual property lawyer I've ever met. And then we've got the CMO, Bruce Forrest, CFO, Bruce Kaufman, Greg Fergus, Paula Keith. So we've got a very, very high level group of people working with Neuvivo. So I'm going to leave it right there and take any sort of questions you might have.
Thank you for that, Dr. McGrath. We do have quite a bit of questions, so we're going to jump right into it. I'm James, and I, along with Grace, will be going back and forth asking the questions that were asked in the chat and the ones that will still be coming. We're going to kick it off with the first question of, do antioxidants help reduce inflammation?
So that was one of the reasons I showed you that whiteboard that had all those little things on it, right? Because it all depends on what you define inflammation to be. I'm sure we've all taken ibuprofen. Ibuprofen works for inflammation, helps headaches, muscle aches and pains. It doesn't do anything to ALS or Alzheimer's. And so it all depends on what your target is. So antioxidants, yes, linopauling would have cured ALS if vitamin C worked. So I'm just saying it all depends on the degree, I guess. So it can work, but does it work for the target you're looking at? That's the question.
Okay, fantastic. Thank you for that answer. Our next question is, what causes macrophages to go quote unquote bad?
It's all related to feedback. Normally for our entire life, our macrophages have behaved pretty well. They keep us out of trouble. They're the immune system for everything below the level of sharks. So there's a lot of species that are below the level of sharks. Above sharks, you get T cells and B cells and things like that. But viruses can do it. So for example, HIV infected macrophages, and those are very long-lived cells. And until an antiretroviral came along that worked, those really drove a lot of the disease. Why a macrophage is abnormal in ALS, I think, is yet to be determined. But you can regulate the macrophages that are abnormal, and that's what we're trying to do. I don't know why they're abnormal to start with, though.
I think it's a great point of jumping off point for future research. Absolutely.
Yeah.
Can results from the SOD1 mouse models be translated to other ALS genetic mutations such as TRDBP?
Yeah, I think what's interesting about the SOD model is that to the extent that you can see the SOD model a parallel that's in humans, then it's absolutely a terrific model. It certainly is robust. I mean, there's no question about it that the animal is relatively easy to care for. You can treat it. You can do daily determinations of the genetic abnormalities as they evolve. And so the TDP43 approach, I think 90% of ALS has got some TDP abnormalities. And I think that's a really good target going after. But that would be trying to get into the central nervous system and figure out how those things work. And we're trying to figure out how to regulate things at the systemic level, and hopefully they feed back into the central nervous system.
And our next question, are there any existing treatments that can block or reduce the production of macrophages?
So there's no such thing as a macrophage-free animal. You know, it's lethal. There are decreasing macrophages. You can make them abnormal, and you can knock them back with drugs. You can't get rid of them really, but they mostly are good. And they're, well, I'll give you a pretty cool example. What's a tattoo? A tattoo is a colored oil droplets that are eaten by the tissue macrophages in your skin. And how long does a tattoo last? You know, and so just imagine if you got a macrophage that doesn't like you very well, and it lasts as long as a tattoo, that could be a problem. So I think that knowing how powerful macrophages are when they decide not to work properly, I think that I think it's an important target for us to go after. And there are companies that are doing it that have targeted macrophages. We've just taken a different approach than others have.
Okay. Our next question, what causes an inflammatory cell to exist?
Well, it's a threat. So the biggest, if the organism is threatened by something, you saw that little picture was a pin wound or a virus, bacteria. So the body recognizes threats very quickly. And then you mount a response against it. And normally we don't feel these threats all the time, because if you think about the colon, we've got bacteria in our colon that help us digest food. And that's a constant threat, because you've got bacteria in there and you don't want that leaking into your blood. And so all along through the colon, you've got this whole wall of immune cells, macrophages, to try to keep all that from your systemic immunity or your systemic blood, from your blood. And so, yeah, that's too much of an answer for that question, but it's a threat.
Simple answers are sometimes the best ones.
Yeah.
What is the role of NF-kappa B in your discovery? And maybe perhaps you could elaborate on it just a little bit.
Yeah, so inflammation, we know a lot about how inflammation occurs and the genes that are regulated in inflammation, because I can take a cell, give it an inflammatory insult, and then measure things that happen. And one of the major drivers of inflammatory response is something called nuclear factor kappa B, or NF-kappa B. And that is a thing that turns on the inflammatory molecules. And if you turn off NF-kappa B, they turn off. So people have tried to make drugs that are just regulating nuclear factor kappa B. Unfortunately, NF-kappa B is required for life too. You know, it's inflammation. It's important, but it's got to be as part of a feedback loop, right? So one of the things that we do with chlorate, and the reason we give chlorate only 3 days a month, is that we want to push the immune system towards a regulation pathway. We don't want to just push it so it's suppressed. We're not being like steroids, for example, and turning everything off. We're forcing it into a situation where it's more functional.
Okay, awesome. So now we had someone very fascinated with your discovery that CRP levels in early markers for COVID-19. How would you correlate your discovery with COVID-19?
Oh, very interesting question. So uncontrolled immune activation is uncontrolled immune activation. And in fact, if you look at the genes that are dysregulated in ALS, they overlap with the genes that are dysregulated in COVID-19. COVID-19, though, is like the monster of immune dysregulation. It's a monster. It's terrible. And some of these effects, like long COVID and those sort of things, where they affect the central nervous system, the brain, those are factors that are probably kissing cousins to the factors we see abnormal in ALS. So it's a matter of degree. You know, you've got chronic problem that's ALS. We don't know what incites it. Probably these molecular abnormalities. COVID is a whopper, comes in there and just blasts the immune system and turns it on. But the factor, it's a matter of degree, I think.
Well, that's fascinating. There's overlap there. Continuing on the topic of CRP levels, could CRP levels be used as a potential biomarker for ALS patients?
Well, the short answer would, I think, be yes. So CRP, so I don't know if I have this slide. I'll just tell you that there's a big study published in from Sweden. Apparently, if you live in Sweden, you have your blood drawn every 3 months, whether you like it or not. And they have this big bank, so they can now look at people as they develop disease, and they have the ability to look back. And so they looked at ALS patients, and they found that ALS patients, when they first presented, had very low levels of CRP. We find that very early on, our patients with low level CRP really don't seem to react to our drug. But as you have the disease a little bit longer, CRP goes up. And that's a common feature. This is a natural history study out of Sweden, as I said. And so I believe the CRP is the primordial immune system saying, we have to fight this. Once that little trigger has been flipped, I think that what we're doing is we're augmenting it with the chloride.
Okay. So now we're going to go back to a little bit of macrophages. Could you please tell us what kind of microbiota is being disturbed due to macrophage activation? And did you see any specific ones in these clinically responsive patients?
It's an interesting question. It turns out it probably doesn't matter. There are, I know that people have tried, well, hopefully none of you are herbalists and selling, probiotics and things like that. But the point is that if you have a leaky gut, it leaks. And so the bacteria almost doesn't matter what the bacteria is. You can try to take different bacteria through your probiotic, but if you have inflammation, it's going to leak into your blood and cause the problems. I mean, that's the simplest explanation. There have been studies done looking at families with ALS patients and with and without ALS. They're all eating the same diet. There are no real differences in the bacteria makeup.
Well, the big question is, when do you start your phase 3 trial?
Well, it's an interesting question. We're planning it, of course, and but we're having discussions with the FDA, and we're doing a couple of things. We're reaching back right now because these studies, the phase 2 studies, were done in 2011, 2017. We're looking at survival. So it's a brand new study, taking all the data, and just like a clinical trial, just saying, what was the survival in this patient population if they had gotten 6 months of drug? So that's our chief medical officer is doing that. That's going to be filed with the FDA. And when the biomarker work, the neurofilament light chain, is also being looked into. So we're looking at things that may help us with moving the bus forward faster, if you will. But we're planning for phase 3, obviously.
Okay. Are there any side effects to the NP1 treatment?
No, there were no SAEs whatsoever.
Well, that's good.
Well, it's turned into water and chloride. It delivers a chloride molecule, but the byproduct is water, so there's no side effect.
Speaking of biosimilar drugs that are currently on the market, are there any that can mimic the new drug for the same purpose?
Well, you can't drink this stuff. This has got to be given intravenously. And it's only stable when it's at a basic pH. If you drink it, you have acid in your stomach. It turns into a poison. Well, relatively speaking, chlorine dioxide. It's used to kill bacteria and things like that, and so you can't drink it. So I think that right now it's formulated as an intravenous infusion to be given over half an hour. You get it 3 days a month. That's it.
Okay. Again, with the macrophages, what are the effects of NSAIDs on macrophages?
Well, NSAIDs interfere with one particular pathway. That's the prostaglandin pathway. They make the thing that makes inflammation and pain not be produced. But that's just so. What's an analogy? I don't want to give an analogy, but the point is that it works for pain relief. The NSAIDs do, but the other kind of inflammation doesn't touch the kind of inflammation we're talking about.
I do want us to be respectful of your time. So because of that, this may be the last question. what are the effects of the non-steroid anti-inflammatory drugs on macrophages?
Well, I think that was the question, last question. I mean, they stop them from producing these prostaglandins, and so your muscles will be less achy and you won't have headaches. But it's just one pathway. There's a lot of pathways that are involved in macrophage activation. Yeah, the NSAIDs are just a small piece and important. Nobody likes to have headaches or muscle aches.
Well,
And
like
my
I said,
time's fine, by the way.
oh, if you're good, then
I
we've
mean,
got
if you
we've got a
wanted to-
few more. We'll keep going for another 5 minutes if you don't mind.
Oh, no problem. No problem at all. I probably overshot how much time I was supposed to be talking anyway. So I'm fine with that.
No, we got a lot of questions, so we'll get through as much as we can in the next 5 minutes.
Okay.
Okay, so our next question then, how are the muscle neuron mitochondria affected in this process? Can access to NP1 be attained today? I think we covered a little bit.
Now, I wish MP001 was available today. I got to tell you, Ari Azir, the CEO of Neuvivo, is superhuman. I mean, with the drug manufacturing, we basically resurrected this drug and are pushing as hard as we can to get it to you all. I mean, this is just it's a labor of love for all of us. Having been doing this for 20 years, and so that's the first thing. I forgot the first question that you said. I was just talking about the availability of the drug. We're trying to get as hard as we can. What was the first part of that question?
The first part was, how are the muscle neuron mitochondria affected in the process?
Oh, the mitochondria. So mitochondria are the body's or the cell's energy machinery. And so you can imagine that if I'm punching you, this is inflammation, that you're going to be consuming energy to try to get away from it. And part of the reason the mitochondria are abnormal in ALS is that you have this terrible assault of inflammation going on, and the cell's trying to resist it. And that's the problem with the mitochondria. And so hopefully, some of these drugs can help the mitochondria fight better. I mean, that would be a good thing.
We had a question specifically about, and forgive me if I butchered the pronunciation of this, taurine.
Taurine.
Yeah, so it's present in energy drinks to the understanding of the individual who asked this question. If it acts as a macrophage regulator and it's in a lot of energy drinks, what are your thoughts about that overall?
There's been a lot of people who looked at that. So if you take a macrophage or a granulocyte, half of the amino acids inside that cell are taurine. And what are they doing there? They're sitting there because this acid is going to come along that's toxic. I mean, hypochlorous acid, the byproduct of inflammation, would dissolve your body. I mean, and that's not acceptable. And so that's why these cells have so much taurine in them. So if you take a drink of taurine, I don't know if you get the same concentration as you would inside that cell. I wouldn't go out and buy a bunch of taurine to drink it, but I think that the taurine clearly, it's an amino acid that lives to block this toxic acid, really, and make this effector molecule. So it's facilitating this whole cycle of immune regulation. And I mean, if I had a taurine company, I would listen to me and go out and advertise it, but I don't know if it's true that you can take it and augment anything.
Okay. All right, well, next question. CRP levels in PD and AD are also high. Will that be a problem for with biomarkers for ALS?
Oh, ALS, CRP is not a disease-specific marker whatsoever. But within an ALS patient population, CRP is going to make it more likely that they'll respond to this approach. That's the whole issue about heterogeneity of ALS. So, for example, just imagine you've got a genetic abnormality that's just driving that disease. It may have nothing to do with CRP, or inflammation. So within ALS, I think CRP is a marker, but it's not a general marker for the disease for sure.
And this will be our last question. Can inhibition of prostaglandins, I always have trouble with that word, actually convert macrophages into inflammatory types and interfere with the resolution of inflammation?
It's too complicated. I mean, the inflammation that's involved with prostaglandin inflammation is different than the kind of information that we're talking about. So they are linked, but they're not. I think it would be hard to manipulate one to affect the other.
Well, with that, I know
So
we're going
that's
to
it then.
that's it for right now. I know we now segue into our non-recorded open forum. You are more than welcome to stick around for it, but no pressure if you've got something else to do to fight off on the research and continue on the fight. I do want to thank you, and I want to thank Grace as well for moderating. And I'm going to turn it back over to McFinn.
Everybody.
Okay, thank you all.
Dr. McGrath, thank you. Your personality makes us all feel like we're part of your family. You're a funny, serious, smart guy, and we really appreciate that you would spend your time with us. I hope you felt that all of us were stepping up.
Well, this is my this drives me.
Okay, well, we're driving with you. Let's put the top down.
Okay.
Step on
All
the
the best. All the best, everybody.
All right.
Take care.
Thank you, sir.
Okay. All righty. Bye-bye.
Bye for now.